Okay. I know you're anxious to get to the parade, so I'll try to finish up early. Huh? Oh, you have lab? Okay. So, okay. So I posted the grades on Moodle, right? And I'll talk to you at the end of class a little bit about the exam, and we'll hand them back. And what I've done, I've also compiled your kind of tentative grades so far in the class, and I'll send you an email on how I calculate that and those sorts of things. So, and what I'd like to do, I think if you're doing, if you have a B, you might want an A, you can come in and talk to me. But what I'm going to try to target some of the students who seem to be having a little trouble, and we're going to try to get you some help. So I'm try to reach out to you, and hopefully you'll come in and see us. So, okay. So what we're going to do today, we're going to finish up on the evolution of multiple loci. And I think this is a nice example that illustrates what are called adaptive landscapes. And it'll also show you what's called a lobster trap, too. So again, this is kind of a classic example of how two genes in bacteria, and remember bacteria, what kind of chromosome do they have? Do they have separate chromosomes, or are they circular chromosomes? They're circular, right? And it turns out all the genes are located together. So they're not located on separate chromosomes, they're all linked together. So bacteria do some unusual things. But this is an example of under antibiotics. It's kind of what we showed before. If this is the ancestral condition, the evolution of resistance is favored, right? And then, so the path is usually going from the small RC to big R small c, and then a compensatory mutation kind of minimizes the negative effects of that resistance gene on growth. And so the pathway is you usually move from this way to that peak and then to this peak. And here, if you have both the resistance gene and that compensatory mutation, it confers a huge advantage in the presence of antibiotics. And the main reason is it confers resistance, but it also, this compensatory mutation increases the growth rate even though that resistance gene is there. So this is pretty easy to understand. The other question is, what happens if you have antibiotics and then you remove antibiotics? What do you think should happen? Should it go somewhere else from this peak to somewhere else? Yeah, you would think, right? This is still pretty high, but is there a higher fitness peak on these four corners somewhere else? Where's the highest fitness peak? Well, it's this one. The problem is you never see that. I will give you an A in the class if you can tell me why. Five seconds. Okay. So the problem is that to get to that point, right, how would you get to that point? You have to go directly over there. The problem is it usually has to go back in this direction to that point or somewhere over in here, right? So let's imagine you lose the resistance gene, right? And it's kind of, but you still have the compensatory mutation. What's the relative fitness of this one relative to that one? It turns out it's lower, right? So the problem is you can never go to any path, you can't go any pathway from here to here because it's lower, right? That fitness is lower. You can't go there because this one still has a fitness advantage. You can't go this way because it has a lower fitness than the big R sub c. The only way to get there is to jump directly across there. And it turns out that's almost impossible to do. And I'm not going to go into all the details. But in your book, what they describe this is, I don't think it's actually the best analogy. They call this the lobster trap. Because once the lobster gets in, it's eating a lot, and then if it tries to get out, what's the problem? It's too big and it can't get out. So it can't go back to it's preferred to this higher fitness state. So I'm not going to belabor this too much, but basically the idea here is when you have antibiotic resistance being favored in these compensatory mutations, you usually stay here, okay? And it's hard to go back to, in the absence of antibiotics, this thing that actually has higher fitness. So it's really hard to do. So you can't go here to here to here. You can't go here over to there. It's really hard to do. back to tomatoes. What do you notice here? Lots of variation, right? And so we see a lot of phenotypic variation in tomatoes in terms of the size, often the color. But one of the things we're going to focus on, the last part of this lecture is to focus on what we call quantitative genetics. So what we're going to try to understand is the heritability of traits that show continuous variation, or what we call quantitative traits, and how do we estimate sort of the genetic components to the variation, for example, in sort of tomato size and any sort of trait that we want to look at. So that's what we're going to do at the end of through the end of this lecture, is look at quantitative traits, how we estimate, how you get them, and then also how selection acts on them. So let's kind of look at it. No matter what we do, what we're looking at is we're always looking at along the x-axis is some trait. It could be, tomato size. It could be height. It could be bill size. Any sort of phenotypic trait that has continuous variation, that you see this sort of often normal distribution. And along here is the number of individuals. So we have a frequency distribution. It's often a normal distribution. Okay. What we're going to say is we're going to characterize that the V sub P basically means what's called the phenotypic variance. So the P represents phenotypic. So what we're saying is the phenotypic variability that you see in a population, whether it's size of tomatoes, length of bills, width of bills, whatever quantitative trait we're looking at, that phenotypic variation is attributable to two main causes. And it's what we call one is G and one is E, where G is the genetic variance and E is the environmental variance, and P is the total phenotypic variance. So all we're saying is the total phenotypic variance in any sort of quantitative trait in a population has two components. There's a genetic component and there's an environmental component. And all we're really saying there is if we look at, let's just say, here basically what we're looking at is here's a population in orange, right? There's the frequency distribution of orange. You can't see it too well because what they're showing in this sort of green color is the overlap between the two populations. But here's another population that has a distribution of that phenotypic trait like this, okay? So what they're showing there are two populations. Does it look like their distributions look different? Yeah, they are different. So let's just go down here and look at individuals that score really low in the trait value and kind of compare them to individuals that have really high trait values. What we're saying is that phenotypic variation that you see in that population is mainly could be attributable to two components, a genetic component and an environmental component, okay? So basically what we're saying is these individuals down here might have particular combinations of genes at multiple loci. Remember, we're not just talking about one gene or two genes, but often multiple genes influencing a trait, right? They have a combination of genes that confers, in general, smaller size, let's say, okay? Do you also imagine that the environment could influence the size or the expression of a phenotypic trait? Yeah. So we can imagine there might be cases where maybe they had limited availability, maybe they had the genes coding for small size, but also there might be an environmental component that they basically had limited access to essential nutrients or water or things like that. So we imagine all these phenotypic traits, both genes and environment, influence the phenotypic expression and the distribution you see in a population. Out here, they might have genes that confer larger size, but maybe they have greater access to resources, more water, nutrients, all sorts of things that also, in addition, might influence what you see in nature. Okay. So what's different? So we're going to come back to that. And basically what we're going to say, what we're going to develop, we're going to look at two kinds of measures of heritability. And heritability basically means how much of that phenotypic variation is due to genetic components, right? So it kind of tells us if you have low heritability, there's not much of a genetic component. If it's really high, you have genes strongly influence the trait. Okay, this is just and I'm not going to ask you to ever calculate this. Broad sense heritability is you take the genetic variance, divide it by the sum of the two, and that tells you, and we'll show you in some sense how we do this. Let's kind of look at these two different graphs. What's different about the orange population and the blue population? What do they differ Yeah, they differ in the phenotypic variability, right? Do they differ in general, you don't have to calculate, do they differ in the average value of the trait between the two populations? Do you think they're about the same? Yeah, they're basically right around here is the average value of the trait, but the orange population has does it have more or less phenotypic variability? Yeah, it has more phenotypic variability, okay? The phenotypic variance would be higher. That measure of that scatter around the average or mean value is much greater. This has a much narrower distribution. The average is the same, and the phenotypic variability is reduced. Okay? What about these two? What? Yep. So here, the average value of the trait is much lower in the blue population and much higher in the orange population. How about the phenotypic variance? Looks about the same, doesn't it? Yeah. And you'd have to you'll do this in lab. But they basically have about the same scatter or variance around them. Okay, so there the average value differs, but the phenotypic variance doesn't differ. And we're going to talk a little bit about what leads to these things when we talk about selection. Okay. So how do we... It took me a while to figure this one out, and I still don't fully understand it reading the book. I didn't read the original paper, but anyway. These are black. This is a bird, primarily Mediterranean. It's in Europe, Mediterranean areas in Europe, France, Italy, other places. It's called the black-capped warbler, and these things do some weird things. And the reason these people were interested in is people were interested in what's going to be the response. These are migratory birds. So certain individuals in certain populations migrate from Mediterranean climates in Europe all the way down to South Africa and overwinter, okay? So they show migratory behavior. Some of them show very limited or no migratory behavior. So they tend to stick around the Mediterranean climates all year round. So there's a lot of variability in terms of whether individuals migrate or not and all sorts of things. Some may migrate short distances in the Mediterranean, but some go really long distances. So these researchers were interested in this, and when you think about climate change and global warming, what do you think is going to be the response in terms of potential migratory behavior? What do you think, what might this bird do less of? Yeah, the idea is you might expect as climate change and global warming, as temperatures go up, they may not migrate as much. They may find that these conditions in terms of temperature are, maybe okay, and they may stick around Europe longer. So they were kind of interested in this from a climate change and a global warming perspective, and they wanted to know what's called the narrow sense heritability. And so I'm not going to go into all the details. They basically collected wild birds from the nests. I think they had about 185 birds that they collected, okay? They reared all these to adulthood, and then they measured this thing called nocturnal restlessness. And I have no idea how that's measured, but apparently you can go in, and at nighttime, I guess when they're restless and they're getting ready to migrate, they're more active at night and they're moving around a lot more. And so that's kind of the signal that they're sort of in migratory, they're going to migrate. So, and again, I don't know all the details of how they measure it, but they measured the nocturnal restlessness. They reared these things to adult. Okay, and then they had also, then they compared the parent and offspring nocturnal restlessness to describe then they bred these captive birds to produce offspring, then they measured these. So the easiest way to measure this is you look at the trait in both the parent and the offspring, right? And that might tell you something about the genetic basis to it, okay? So they took the parents, then they bred them, then they produced the offspring, and then they measured. They did this all in the lab, okay? Here's what they found. And again, the way to look at this is along the y-axis is what's called offspring nocturnal restlessness. That's from these individuals. Each point represents the average value of the offspring and then what's called the mid-parent. So the mid-parent is just the average value of the male, the father and mother. So it's the average value, mid-parent is the average value of that offspring's mother and father. Okay? And then each dot represents each parent-offspring combination. Okay. So when you look at this, what is it, when you look at this line, what does it kind of show you? Does it look like they're kind of related? Yeah, you can see, what's this line represent? Have you done regressions in lab? Okay. This is what's called a regression, where you look at the relationship between the offspring value and the midparent value. Does it look like it's kind of related? It's going up, okay? So that in general, parents who have low nocturnal restlessness give rise to what kind of offspring? Ones with low nocturnal restlessness. And as you know, parents that are highly restless at night, in general, give rise. It's not, is it perfect though? Does it look like it matches it exactly? Not really. So it turns out what you can measure is what's called the slope, which is the change in y for every change in x. And it turns out this is what we call the heritability, narrow sense heritability. Turns out this value in this graph is about 0.45, okay? And I forgot my iPad, so I'm going to have to do something else. Do I have to go to the board? Okay, is that okay? Okay. So let's look at, do I have chalk? No. Okay. Is it in here? Oh, here's some. Okay. So let's put offspring here, midparents. I'm not going to put all the dots up there, but what if you had something where something looked like that? Would you say that's a heritable trait, that the offspring values are very highly correlated with the parent values? Nope, not at all. In fact, that slope equals 0. Okay? So there's no correlation between the offspring value and the midparent value. So would that be a heritable trait or not? Turns out that's not heritable at all. So this h squared, which is the heritability, would equal 0. Okay? Let me show another one. So the one there is 0.45, right? So for every change in x, y goes up an amount, a corresponding amount. Parents. And let's say you never see anything like this in nature, but let's imagine you had something like that, and you don't know the scale and everything, but this would be a case where the offspring values are almost exactly correspond to the parental values. What do you think the h squared would be in that case? Yeah. So that's going to be the case where that slope is equal to 1. Do you think that's a very heritable trait? All the phenotypic variation is due to genes. So that's basically what that's saying, is that's a highly heritable trait. And what that says is all the phenotypic variation that you see in the population is attributable to genetic differences among the individuals. Okay. Okay. So those are kind of the two extremes that you can get. Okay? And this case kind of showed 0.45. Okay. So the idea here, the take-home message from this is that this is a heritable trait. There's a lot of phenotypic variability in nocturnal restlessness and presumably migratory behavior, right? And the idea is it's pretty heritable. 0.45 is not bad. There are some environmental influences on it, but a fair number of genetic components to it. And the idea here is that if selection were occurring, migratory behavior could evolve, is the take-home message. So the key is you need sort of genetic variability for selection to act on a trait, right? Remember we talked about that the last time, or we've talked about that quite a bit. So you need that heritability for selection to be able to cause change in a trait. So we think that this thing could evolve and migratory behavior could shift. And in fact, what a lot of people are seeing is that migratory behavior is changing pretty dramatically in response to, in some cases, warming patterns. Okay. So the other thing is, the last thing we're going to focus on, we'll focus a little bit on this. I'm not going to ask you to calculate any of these things, but one of the things we're really interested in is when selection acts on a quantitative trait, how much change do we get? So how much evolutionary response? So R is what we call the selection response. And what we're saying is it's influenced by two factors. The thing we just did, which is H subscript, that should be H. That should be up there, but anyway, okay. That's the heritability, right, that we just measured, the narrow sense heritability. And S is what's called the selection differential. And we'll be going through an example of this. So let's imagine, and you're not going to have to calculate any of these, but we'll kind of go through this. Here's the parental generation before selection. And let's just say we have some value of some trait. And I think what they're primarily looking at here is color, okay? So we have some measure of pigmentation or coloration in the parental generation before selection. The average value in the population is 0.6. Then there's a selection event, and those three individuals die, and then these give rise to offspring. Okay, so this is the parental generation after selection. Here's the offspring generation after selection. Okay. Let's look at how we determine what's called first the selection differential. I'm not going to ask you to do this, but the selection differential is what's the difference in the average value of the trait in the parental generation after selection minus the parental generation before selection. So it's kind of telling you how much has the average value of the trait shifted just before the parental just within the parents. And so what it's saying here is it's shifted from 0.6 to 0.8, so the value is 0.2. So there's basically been about a 20% increase in the average value coloration when these things died, okay? Now the next thing that has to happen is the heritability, right? They just died. You don't know if that, how much of that is influenced by genes or environment. And then here's the offspring generation afterwards and before selection, okay? So basically, you can then figure out R equals this value, O sub 0, right, 0.72, minus the parental value. And it's shifted, the response has gone from 0.6 to 0.72, so it's gone up 12%. So you'll notice there's a little difference here, isn't there? What do you notice here? In this parental generation, it went up 0.2, but in the offspring generation, it only went up 0.12. So something clearly we need to know something about, and that's determined by the heritability. Okay, and again, I have to go back to the board. And so let's kind of look at this. And you can actually determine from these values what the heritability is of this trait. Let me ask you something. Do you think the heritability of this trait was 1 in this case? Anybody want to vote for that? Or do you think it's less? Turns out it's less, okay? So let's put R equals H squared S. We've solved for 2 of those, right? So R is equal to, it responded 0.12. H squared, and then S equals what? 0.2. So what does H squared equal? H squared equals 0.12 divided by 0.2, which is 0.6. Okay, and again, you're not going to have to do this, but the key here is that basically what this shows is this thing shifted in one generation from 0.6 to 0.72. Okay, so it shifted. So there's been a response to selection. And there's some heritability there, so it shifted. The genes for, coloration, increased coloration, darker coloration influenced that trait, and it increased in frequency. So the thing shifted from 0.6 to 0.72. If the heritability were 1, what would the outcome be? What would this value be? Yep. So if this were to occur, if this were an highly heritable trait, this would actually be 0.8. It would be 1. Okay. So that's what would happen in that particular case. Okay. So let's look at an example of this that really, I think, illustrates this really well. So in artificial selection, in corn, we're really interested in a couple things. One is people are interested in kind of the oil content, okay, and how much oil you can get out of corn for various sorts of things. So this is an experiment that was started basically at the start of the experiment in 1986. So this is sort of the ancestral condition. The oil content ranged from about 4 to 6%. By 1998, the population, they went in both directions. They selected for low oil content, and then they also selected for high oil content. By the population selected for low oil content had this is the one for 1986. By 1998, the population selected for low oil content had gone down. So there was clearly a response to selection. So you can go to low oil content. Most people aren't interested in that, okay? Don't want low oil content in their corn. They want much higher oil content. Here's what happened by 2004. Does it look like there was a response to that artificial selection? So it shifted from about 4 to 6%. The population selected for high oil content had gone to near 20% or above, okay? So clearly there was a strong response in terms of oil content to this artificial selection that was occurring. And all they were doing, what were they doing each generation in the starting population? In each generation, they were selecting those extreme individuals with oil content to lead to the next generation. Okay. So I'm not going to spend too much time on this. Is a but what this shows is that here's what happened. Here's what happened in year 1900 plants, then the selected year, and the discarded plants. So let's look at. Here are the 1899 plants. Those were the oil content that was selected to found the next generation. And so they chose individuals that are above 6, around 6.6 in terms of oil content, okay? So this is the selection differential between the mean of the selected ears and the mean of the entire. So it went from, average value went from about 5.6 to around probably about 6.2. So they selected extreme individuals. So they selected those really extreme individuals, okay? And then they measured what was the average value of the trait in the 1900 plants. And what they showed is there was a response. The selection response is the difference between the population means in 1900 and 1989. So it went up, okay? Did it go up as much as the selection differential? Not as much. So, but it still is a heritable trait, okay? So the key here is that you can impose strong selection on a trait. If there's a heritable component, if there's a genetic basis to that variability, you're going to get a response. How heritable it is determines the magnitude of the response. If it's highly heritable, you get a big response. If it's not heritable, you get no response, okay? And this really illustrates the fundamental theorem of natural selection, right? That you need genetic variability for a trait to evolve and change, okay? And we're just applying this to quantitative traits, okay? This next slide, I think, is the key one. And this is going to lead into all sorts of things in terms of speciation, population divergence, and these 3 kinds of selection. So we're going to talk about 3 kinds of selection on quantitative traits. And we're not really getting into heritability and all sorts of other things. We're going to ignore that right now. But here what we have is, here we have sort of the number of individuals before selection. So here's the ancestral population, any sort of trait, any sort of phenotype we want to look at. And basically what we're saying is these all differ a little bit, but they're all pretty much the same in terms of the starting population. Okay. So what we're going to contrast is what's different about directional selection, what's different about stabilizing selection, and what's different about disruptive selection. And what we're really going to focus on is what effect do these three different kinds of selection have on the average value of the trait and the phenotypic variability in a population. So this is what we call directional selection. Why do we call it that? Which individuals have the highest fitness and which ones have the lowest fitness? Well, does the average have the highest? So the average value in the trait is around 7. Yep. So what we're saying here under this kind of selection, those individuals with the most extreme value of the trait have the highest probability of surviving. And it's kind of a linear relationship, right? And what about the individuals that are around 1? What's their probability of surviving? Extremely low, right? Okay? So what we're saying is this is what's called directional selection. There's usually some sort of linear relationship between the fitness for survival or reproduction and the value of the trait. This is positive, right? This is a positive directional selection. Can it go a different direction? Yeah, it could go that way. But anytime there's sort of a linear relationship between fitness is always going to be along here, so we're saying survivorship, and the value of the trait. Okay? What's stabilizing selection? Which individuals have the highest fitness and which ones have the lowest fitness? Yep. So individuals with the extreme values of the phenotype, down here and down here, have the lowest fitness. Which individuals have the highest fitness? The intermediate or average. So we'll come back to that. Here's another way these are all what we call fitness curves. So they measure the relationship between fitness and the quantitative trait. What's different? What about this one? How's this one different from that one? Yep. The two extremes have the highest fitness, and which phenotype has the lowest fitness? The intermediates or the average. Okay. So selection favors the extreme, intermediates, and two extremes. Now, what effect does this have on, so here's the distribution of the trait before selection. Here's the distribution of the trait after selection. What do you think's happening here in terms of the average value of the trait and the variability of the trait? What do you think's, what's happening to the average value of the trait under this kind of selection? Think about the oil content, right? That's basically what we did in the oil content experiment. That's what we do in all artificial selection measures. We usually select the most extreme individuals, and we expect a response in that direction if it's heritable, right? So this shows you that, in fact, it's hard to see, but there's the average value before selection, there's the average value. So it went up a little bit. It went up at about 7.5, for example, okay? We'd want to know the heritability if we really wanted to know, but okay. Where do you think that's eventually going to lead? Huh? Yeah, you might expect here that over time, if you do this for a lot of generations, you might lead to extreme individuals, okay? What do you think's happening to the variability of that trait over time? Is it going up, going down, or remaining the same? What's a prediction? Yeah, it's going to go down. So directional selection does two things. It favors a more extreme phenotype, so it changes the average, and it reduces phenotypic variability. Okay? How about stabilizing selection? How is it different from directional selection? So just think stabilizing. Basically, what's happening to the average value of the trait when the average phenotype has the highest fitness. It stays, it's not changing. So it's stabilized, it's stable. So the idea here is, and this should be under this kind of selection, the average value of the trait remains exactly around 7. What happens to the phenotypic variability? What are you getting rid of? These extremes. So you're basically cutting off these extremes. You're narrowing this distribution. So the mean remains the same, and the phenotypic variability is reduced. The average value around the trait is reduced. Okay? Now, last one is disruptive selection. What's happening? This has to be a perfect curve to get this. What's happening to the average value of the trait under disruptive selection? Yeah, basically what it's doing is it's actually that average value of the trait is not changing, is it? It's actually right still at 7, but what's happening? What's happening to the phenotypic variability? It's going up. It's increasing. You're seeing fewer of these individuals that are intermediate and a greater representation, not the greatest, they didn't do this very well, but anyway, more individuals in the extreme. Okay? So here the mean remains the same. What about the phenotypic variance? Going up. Okay? Where do you think this is eventually going to go? Yeah, so if you had to think about which of these two mechanisms lead to populations or things becoming different from one another out of these three? Disruptive selection will lead basically to an extremely small phenotype, right? And an extremely large, whatever that phenotype is. It'll lead to the two extremes. This thing, can this lead to divergence? Yeah, and so it can lead from the ancestral population experiencing a different kind of environment or different kinds of selection can lead to also populations becoming different. Okay, does this result in much evolutionary change in the average value of a trait? Stabilizing selection doesn't really do it. It just kind of stabilizes things. Okay, so as we come back, as we start getting into speciation, we're going to be talking a lot about these two kinds of selection as drivers of species formation. Okay. So on this one, I'd really know how the change in the mean and the variance of a trait for each kind of selection and how they're different. Okay. Let's do an eye clicker. Let me